Respiratory Division, ICMR, National Institute of Epidemiology. I know I present to you earlier, but you aren't here. IC Indian Council of Medical Research, Chennai. He was awarded doctorate from in microbiology and virology from University Jadavpur University, Kolkata. Previously worked at National Research Center of Disease Control and ICMR National Institute of Cholera and Enteric Diseases. He published 41 research articles, 2 books and chapters, and 1 book. And he was selected for Infectious Disease Fellow Program at American Society for Microbiology Conference, Microbe 2018 at Atlanta, USA during June 7 to 11, 2018. He is a principal investigator for DST-JSPS Joint Research Project under prestige Indo-Japan Cooperative Science Program. And he is recently made an exchange visit to Professor Nobumichi, Professor and the Head Department of Hygiene, Sapporo Medical University School of Medicine, Japan, between 26 January to 11 February 2020. So I would like to welcome you, Dr. Bala, to start sharing your screen now, please.
Yes.
Yes, I see.
Good evening. Actually, am I audible?
Yes, you can raise your voice a little to hear you clearly. I hear you.
Is it audible?
Yes, you can raise your voice a little.
Oh, let me increase. Yeah, good evening, everyone.
Yes.
So I'm very thankful to Dr. Bala for inviting me for this presentation in this first international webinar related to COVID. Shall I continue?
Yes, please continue.
Yeah, okay.
Okay. You can take your next slides. I think Dr. Bala.
Yes, I have come now.
Okay, it's okay.
It's ready now?
Yes, I hear you. I hear you.
Yeah,
now.
okay. Yeah, I'm starting.
You can.
I can share the screen?
Okay.
Yes?
Yes, it's okay.
Yeah, okay.
Go ahead.
Thank you.
You are welcome, dear.
So, yeah, this is the National Institute of Epidemiology. It's an Indian Council of Medical Research Organization in Chennai. I'm working here. So I acknowledge this institute for my current affiliation. So let's go into the topic, novel coronaviruses. Actually, there is a list of experts and research scientists who have presented almost on all aspects of the novel coronavirus. Though I like to give a final conclusion because I am the last presenter here in this webinar. So in coronaviruses, there are a group of viruses that have crown-like spikes on their outer surface. So corona means in Latin, it is crown. So hence it was named as coronavirus. Currently, there are 7 human coronaviruses types were identified that infect humans, causing diseases ranging from common cold to fatal or severe infections. Among them, 4 human coronaviruses, including human coronavirus 229E, NL63, OC43, and HQU1 are sporadically attributed to infections similar to common cold, but in rare cases can cause serious infections in infants, adolescents, and elderly population. The remaining 3 coronaviruses comprising SARS, that is severe acute respiratory syndrome coronavirus SARS, Middle East respiratory syndrome coronavirus MERS, and the recent one to get into this list is severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, can affect the lower respiratory tract and cause a severe disorder and pneumonia in humans. So I have just made a table where it deals with the epidemiological and clinical characteristics of SARS, MERS, and SARS-CoV-2, this is COVID disease. So in general, the year of origin of SARS virus is November 2002. Whereas MERS it is June 2012, and COVID-19 by mid of December 2019 in Wuhan city, Hubei province of mainland China. The etiology, the causative agent is SARS coronavirus for SARS, MERS coronavirus for MERS, and SARS-CoV-2 for COVID-19. See, the original location of this infection, first SARS was in Guangdong province in China, MERS was in Jeddah city in Saudi Arabia, and COVID-19 was originated in Wuhan city of China. So there were a number of confirmed cases for SARS was around 8,098. MERS was around 2,200 odd, whereas the COVID-19 is more than 20 million. Similarly, the mortality rate was 9.56 for SARS and 35.37 with respect to MERS, whereas COVID, though it's cost spread, it is only 3.68%. This is the data which we have taken from WHO COVID-19 situation report as on yesterday, 12th of August. So the number of countries affected by SARS is 29 countries around the world, whereas MERS affected 27 countries, and COVID-19 has spread across the world and caused more than around 216 countries and territories across the globe. The natural reservoir host, in general, we were thinking about the bats, whereas the intermediate host, which plays a main role in transmitting from the reservoir host to the humans. For MERS, it is civet cats, and for for SARS, it is civet cats. For MERS, it is dromedary camel, and pangolin is still doubtful. There is no proof yet. And the genome length of SARS and MERS and SARS-CoV-2, it is 27.9 kb size for SARS and 30.1 kb size for MERS and 29.9 kb size for the SARS-CoV-2. So the genus, it is beta, SARS-CoV belongs to beta coronavirus lineage B. This MERS belongs to beta coronavirus lineage C, and SARS-CoV-2 belongs to beta coronavirus lineage B. So the incubation period is around 2 to 14 days, whereas for SARS it is 2 to 10 days and 2 to 14 days for MERS, as well as 1 to 14 days for SARS-CoV-2. The basic reproduction number, we say R0 value, it is 2 to 3 for SARS and less than 1 for MERS, and it is around 2.2 for SARS-CoV-2. Transmission, the main way of transmission is through the respiratory droplets and the close contact with the diseased patients. It is common with all the 3 viruses. Human-to-human transmission is very efficient in SARS, and it was limited with MERS, whereas it is possibly more efficient among SARS-CoV-2. That's why we could see a number of cases increasing on day by day. The predominant cellular receptors for the adhesion of this SARS virus with human, this thing is ACE2. For MERS, it is DPP4, that is CD26. And for SARS-CoV-2, it is ACE2. Transmission region, it is globally spread. SARS and SARS-CoV-2 has spread globally, whereas MERS has regionally transmitted. The pandemic potential, as we know, SARS also has reached pandemic, and SARS-CoV-2 is currently the current hot topic, and it's a global crisis pandemic. Whereas MERS doesn't have a global pandemic potential. The common signs and symptoms include fever, malaise, cough, and shortness of breath for SARS-CoV-2, whereas shivering and cough and shortness for SARS, and diarrhea and cough, shortness of breath for MERS. The major complications includes pneumonia, severe acute respiratory distress syndrome. If not properly treated, may result in death. So the current diagnosis, what we are using for the suspected patients, are the real-time PCR, which detects the virus from the nasopharyngeal and oropharyngeal swabs. And for surveillance purpose, we are using antibody-based serosurveillance purpose. So we are doing antibody-based detection. IgG for the past infection, IgM for the current infection. As far as treatment is concerned, for SARS, it is glucocorticoid and interferon were given. And for MERS, it was ritonavir and lopinavir. And similarly, for SARS-CoV-2, there are plenty of drugs on clinical trial, and even vaccines are also on trial, but still on lab base. We have recently seen a report from Russia, which they say they have made a vaccine and currently being introduced for the use of public. Currently for SARS-CoV-2, the lopinavir and ritonavir under clinical trials and on use. So this is with respect to the genome of the SARS-CoV-2, the schematic representation of the SARS coronavirus 2. It has a spike protein on the membrane, the nucleic capsid, the envelope, and the single-stranded RNA. It's a nucleic acid is a single-stranded RNA virus. So epidemiology. So it is in general, we got the information that the spread, the origination of the virus was in December, mid of 2019, hence it was named a 2019 novel coronavirus. It was identified as a cause of pneumonia of unknown origin or anonymous etiology in Wuhan City, Huanan, a seafood wholesale market, Hubei province of mainland China. On 7th of January 2020, WHO provisionally named the infection 2019 novel coronavirus. And it was subsequently renamed as SARS coronavirus 2 by the Coronavirus Study Group of the International Committee on Taxonomy of Viruses, ICTV, on 11th February 2020. The disease caused SARS-CoV-2 was called as coronavirus disease, COVID-19. On 30th of January 2020, the WHO announced that the COVID-19 outbreak to be a public health emergency of international concern. And on 11th of March 2020, the WHO declared COVID-19 as a pandemic. As of 12th of August, as of yesterday's report by WHO COVID-19 situation report, the number of countries affected were 216, were infecting more than 20 million cases, and more than 7 lakh deaths were reported, as per situation report 205. So if you deal with the pathobiology of the novel coronavirus, SARS-CoV-2, it has a Hess-glycoprotein on the surface, which is used for interacting with the host cell receptor and activates the entries of viruses. For beta coronaviruses, the receptor binding domain of the S protein arbitrates the binding with the host receptor. And the ACE2 receptor, that is angiotensin converting enzyme 2, are well-known host receptors for SARS-CoV-2, which makes use of ACE2 to enter into the host cells. In the respiratory tract, ACE2 is broadly expressed in the epithelial cells of the bronchi, trachea, alveoli, alveolar monocytes, and bronchial cirrus glands and macrophages. The viruses replicate in these target cells, and then mature virions are released from the primary cells and infect new target cells. So this is a schematic representation which was recently published in one of the paper. So that's why we are just showing you this. The entry of the virus, the entry into the virus, and then protein translation, and becoming new virus and being released. The symptoms and complications. The main symptom of COVID-19 are cough, shortness of breath, fever, and sneezing. So by this way, the virus become a droplet and it spreads to other people. The infection spread. Transmission is through droplets and aerosols, and even direct contact with the droplets or on the surfaces. Contact surfaces, it transmits the infection. The main complications may contain throat pain, pneumonia, acute respiratory distress syndrome, and a lung infection. SARS-CoV-2 is spread very rapidly from one person to another through direct contact, respiratory droplets formed due to sneezing and coughing, and intermediate fomites from both asymptomatic and symptomatic cases during the incubation period. Recently, we have seen a report from WHO where it doesn't transmit. Asymptomatic cases may not involve in transmission, but it's to be analyzed everywhere. It changes for conditions. The incubation time for COVID-19 is approximately 1 to 14 days. The role, the R0, that is the reproductive ratio or reproduction number, is actually the number of transmissibility from one infected person to others. How many person he can transmit the infection is denoted as R0. And for COVID-19, it is 2 to 4 based on the reported this thing. So diagnosis, the modern diagnostic tools for COVID-19 are molecular techniques, viral culture, and serology. The general investigative approach is a molecular detection method such as real-time reverse transcription polymerase chain reaction, and then real-time reverse transcription loop-mediated isothermal amplification, RRT LAMP, and reverse transcription PCR, reverse transcription LAMP, by analyzing the nucleic acid, that is RNA isolated from samples of respiratory tracts such as sputum, nasopharyngeal swab, deep tracheal aspirate, or bronchoalveolar lavage. The identification of novel coronavirus is based on previous travel history to COVID-19 affected countries. Primary and secondary contacts of the laboratory confirmed COVID-19 cases, and the laboratory confirmed COVID-19 cases, those who were without any of the above conditions, above signs and symptoms. We say asymptomatic cases, but having previous travel history to COVID-19 affected countries, or even no history of contact with COVID-19 confirmed case are termed community transmitted cases. Yeah. Okay, shall I continue?
Sorry for interruption.
Okay, no problem.
There's some noise, but it's sorry, no.
Okay.
It's okay.
Oh, okay. Also, the vaccine with respect to the current available or the R&D research and development with related to vaccines. A vaccine as a successful vaccine is very necessary to break up the chain of transmission from animal to human or human to animal. But anyhow, we need to stop this spread of infection. So vaccine plays a main role, but we are still on doing research and development. It is essential for antiviral treatment in the control of epidemics or pandemics caused by these emerging and re-emerging exotic and highly infectious viral etiologies. Now, the whole world, it become very small and people hence travel a long distance in a very short duration. So if a person gets an infection, he can easily transmit that infection, or he can easily get an infection from anywhere in the globe because people are moving around. So globally, the research institutions and pharmaceutical companies are spreading up their research and development for developing a potential candidate vaccine against coronaviruses. The improvements of a safe, secure, and successful vaccine, effective vaccine against SARS-CoV-2 for non-immune persons are very urgent necessity now. So which plays an important role in controlling of the ongoing disease transmission. People, we say the other comorbid condition, people with diabetes, long-term chronic medications, those who have hypertension, on other cardiovascular diseases or malignancies, they are more prone to get severe infection, which may lead to death. So, to affect it, to prevent it, so we need to work to get a better way. And treatment and therapeutics, the most effective way for the treatment of COVID-19 is to investigate whether existing antiviral drugs are efficient. Because making a new drug to come to the market for commercial purpose will take 10 to 12 years. So we should manage with the currently available drug, which we can use it again. The currently established drug for one treatment of one infection, which can be used, that we say repurposing of drugs. The currently existing drugs, we should try to see the uses of the currently existing drugs to work against this SARS-CoV-2. In earlier epidemics of beta coronavirus, many antiviral drugs like interferon, darunavir, cobicistat, ribavirin, lopinavir, ritonavir were investigated with a few confirmed promising in vitro outcomes. So at present, the in vitro antiviral efficacy of accepted drugs such as penicillin, ribavirin, chloroquine, other chloroquine, and many other things of two broad range antiviral medicines like excuse me, favipiravir, remdesivir for COVID-19 therapy is under trial now. So among the investigated drugs, both chloroquine and remdesivir were observed to be better efficient in treating for COVID-19. Similarly, interferons, thymosin, and intravenous gamma globulin were considered to improve our immune system to combat SARS-CoV-2. Researchers earlier proved that the protease inhibitors such as ritonavir and lopinavir, drugs for human immunodeficiency virus, HIV antiretroviral drugs, also could enhance the results against SARS-CoV-2. An easy but very effective treatment approach is currently carried out using convalescent plasma, a serum, from patients who have recovered from coronavirus infection and to treat very severely infected cases, still on experimental way. Conclusion, researchers are currently working on various places around the world in developing an appropriate, safe, and successful vaccine candidates and antiviral drugs for prevention and control of the deadly novel coronavirus disease. The outbreaks of SARS-CoV-2 propose that coronaviruses can cause enduring threat to human life even at the modern era. The best hope we can rely is that the outbreak will ultimately come to a control and the population's immune memory of the coronavirus, that is herd immunity, will naturally control further spread in the community. But we should not forget the lessons learned from this pandemic, and we should be prepared well in advance, precisely act and initiate the appropriate preventive measures for the next wave of outbreak of coronavirus, if it is going to happen, or any other exotic viruses in the near future, if necessity arises. So these are some of the research publications. We have made a review article which published in the Pathogens Journal. The how to cite is given. Coronavirus disease challenges in the global perspective. With the DY number we have given down. And this is one of the review which is got published in Health magazine called Health for the Millions, under the issue of communicable diseases, emerging threats, and strategies for sustainable management. We have written one article on this. So thank you for your patience for listening to this presentation. If you have queries, I'm open. I have given my email ID and mobile number. If you have any queries, you can contact me as well. But right now, if you have any query, you can. I'm ready to answer. Though we have unmute your call. Dr. Wala?
Okay.
Unmute, yes.
Okay, thank you.
Okay.
I need your last slide presentation, last slide.
You need the last slide?
Yes.
Oh, sure. Okay. You mean this one?
Yes, I mean this slide. Where is this slide?
Okay. This was taken at Nagoya. It was in Japan, Nagoya city. It was an international convention center where the person who is giving pose is me, and the person who is taking the snap is Professor Shyamal Pal. You could see the samurai standing, and the statue is a samurai.
Yeah.
But
In
you
the
could see the
background.
reflection. You can see the reflection. The mirror image only has a sword, whereas from this angle, you couldn't see the right hand of the samurai.
Yeah.
He's having his sword on the right hand, which is reflected in the mirror, but not on the statue.
Yeah, because I wonder it's not in India, and
No, it's not in India. It's in Japan.
background.
I visited. Yeah.
Yes, nice presentation. Thank you so much for accepting
Okay,
my
sure, sure.
invitation, my dear
Okay.
Professor Dr. Bala. And
Yeah.
I am so honored to have you today because when I visited Chennai in
Chennai.
yes
Yeah,
in
yeah.
in
Indo-African
November.
collaboration. We have done,
Yes,
yes.
last November 2019 to take a training course in NIE. It was an amazing experience. And NIE really is a unique institute dealing with epidemiology and really care about the Indian population and the Indian problems. And all days in the course, I found all professors and lecturer was caring about teaching us all things because and accepting many questions from all of us in the course classes. So thank you, and I'm looking forward to visit India soon, especially in IE and ICMR because I am.
Yeah, we also hope if the pandemic ends, we also will visit.
And you have to visit
Yes,
Egypt also.
yeah, sure. I'm willing.
Yes, you have visited Egypt also. I'm so happy, and I can accept any question now for Dr. Bala, and sure for Dr. Hisham after Dr. Bala. So, any questions? So I have a question.
Yeah, please.
Okay, regarding to epidemiology and COVID-19 in India, I hear in news is a large number of every day increasing, and India didn't reach till to the plateau or to the top of this COVID-19 crisis. So, what is the measurement? I know India is the largest population, and that is reflect so badly about increasing numbers. So what your opinion?
Yeah, actually, India is the second largest populous country in the world, and we have different. We have very huge diversity among region, people, their culture, many things. And then we have more than 36 states and union territories where people are being covered. The health system is covering every aspect. And ICMR is taking a lead role in distributing the kit and doing the supporting the supplying with the logistics and doing the testing aspect. People still they don't want to openly come and tell that they are getting some infection. They manage with the drug whatever they have. So here we have both allopathy, Ayurveda, Siddha, Unani, homeopathy, many other forms of medicines are being practiced. So people, those who have infection, they have got some stigma. So initially people were due to this, what do you say? There is a stigma still. So if anybody is having COVID positive, immediately they will be locked down and their road is being blocked and everybody are facing one or the other issue. And there were many other issues also coming up. But still, ICMR is providing, and we are increasing the labs daily, both private and government health facilities. We are doing testing. The number of tests per day is increased up to 10 lakhs. Recently 1 million tests we have done throughout India. So when you do the test more, and then we are getting results also more, but thereby people recovering and getting to home also it is getting increased. Whereas these people like the comorbid condition people, they are getting severely affected, and we are still hoping. Since we are also a developing country, we have to manage with whatever facilities we have. And the lockdowns, the quarantine, everything really worked. So there was good response from the general public as well, not only from the health sector, even from the general public also. That's why we are still lagging. We didn't reach the peak. So earlier there was a prediction model where we thought by the end of July, it should have reached peak and then started declining. But still, due to this other mitigation, other implementation, other things. So we are still dragging. Anyhow, once it reaches peak, then it has to go down. The pandemic, the normal curve should come. So, plateau should reach, and then we should also get free that the condition, every condition turns well. All the schools and colleges are still closed. There is only health care workers and other people can move. Otherwise, the schools and colleges are closed, and we are still. The normal situation has to come. Everything, anyhow, we should manage it.
I wish all these crises and pandemic all over the globe, especially yes, especially developing country and my country, your country, can cope with this pandemic crisis. And I hope India reaches the bottom of curve and decreases the case soon. Thank you so much, Dr. Bala, and may